Welcome to a set of tutorials on the visceral motor system. I'd like to take this set of tutorials in three parts. This first part, I'd like to give you a bit of an overview, including some discussion of the functional divisions of the visceral motor system. And then we'll have a session on the hypothalamus, a very important part of the brain's means of controlling visceral motor activity. And then lastly, I'd like to illustrate some of the principles of how visceral motor activity is integrated with sensory input, as well as somatic motor output in the governance of one important function, and that's the function of controlling the bladder. Well, this topic of visceral motor integration and visceral motor control relates to some of our core concepts in the field of neuroscience. You're very used to hearing me assert that the brain is the body's most complex organ, but that is a fact that I hope is not lost on any of us as we pursue a deeper understanding of how the brain governs the body within which it resides. We'll also be talking more about circuitry today that is genetically determined, but still subject to life experiences and the modifications that use can feed back into a functioning nervous system. Alright, so in this first part of our tutorial on the visceral motor system, I have three learning objectives for you. First, I want you to be able to describe the anatomical organization of the sympathetic and parasympathetic divisions of the visceral motor system, including the sources of preganglionic innervation and the location of the postganglionic visceral motor neurons. So, as this objective implies, there are important differences between these two major divisions of the visceral motor system that I want you to be able to have a discussion about, perhaps with a friend, even a family member who may be curious as to what you're learning in medical neuroscience. I want you to be able to characterize the major functions of the sympathetic and parasympathetic divisions. Now, that's not to say that I want you to account for sympathetic and parasympathetic governance of each visceral motor effector system in the body. That would be beyond the scope of this course. But rather I want you to be able to have a discussion that highlights the general characteristics of sympathetic activation, parasympathetic activation. And one great way to do that is to pick out a subset of those visceral motor effectors that are particularly instructive for you, and think about how these two systems interact to govern the activity of that system. And then finally, I want you to be able to identify and discuss the neural centers in the central nervous system that regulate the outflow of activity in the preganglionic fibers of the visceral motor system. So we want to back up to higher centers in the brain and understand how does the brain govern the coordinated output of these two divisions of our visceral motor system. Well, let's now begin by considering in broad overview the functions of the visceral motor system. Well, what the visceral motor system does is it maintains the internal environment of the body, and it regulates the activity of the visceral organs, the glands, and the blood vessels. The visceral motor system governs circulation, respiration, digestion, metabolism, bodily secretions, thermal regulation, and reproduction. Elements of this system, together with components of the somatic motor system, are involved in the experience and expression of emotional behavior, and indeed other dimensions of cognition that have an impact on the internal workings of our body. Well, I think you're familiar with some of the basic functional divisions of the visceral motor system, and perhaps I should pause here and just state that I've been using the term visceral motor system. You may be wondering, well, aren't we talking about the autonomic nervous system? And indeed we are. These terms, for the most part, are synonymous. I prefer visceral motor system because it allows us to focus on the effector systems that we're concerned about in this particular session, that is, our visceral organs. There's also an element of autonomic nervous system function that involves the somatic motor system. So I don't want us to conflate thinking about the viscera with thinking about somatic motor effectors. That's a point we'll come back to in the third part of this tutorial. Just so that you're not thrown off by my terminology, so from here on out I'll be referring to the visceral motor system as those aspects of autonomic function that pertain to the governance of visceral effector systems. Okay, so with that said, we basically have three functional divisions of our visceral motor system. We have a sympathetic division, which is involved in energy mobilization. There's a parasympathetic division, which is involved in energy conservation. So already you get a feel for the complementary actions of these two principal divisions of the visceral motor outflow. But there's also a third division that I'll just touch on very briefly, and that has to do with the neural networks that govern the activities of our gastrointestinal tract. We call this the enteric division. So this is a system of neurons that function in something of a semi-autonomous way. I'll say more about this system in just a minute. And what this system does is it coordinates our digestive reflexes. Well, again, to think in very broad, big picture terms, the coordinated activity of these three divisions of the visceral motor system is really all about two principal kinds of functions. One is homeostasis. So this is a wonderful term coined back in the early part of the 20th century by a great physiologist by the name of Walter Cannon. And what Cannon had in mind with homeostasis is the constancy of the internal environment. That is, working to keep the body more or less at a set point of some sort. Well, homeostasis is what our bodies are about, arguably most of the time. But we couldn't survive very long if this was the only mode of action of our visceral motor system. Indeed, there are times when, in order to restore that proper balance, we have to change. We have to push the body into rather emergency action. And so this concept is what we might have in mind with this term allostasis, which was coined more recently by a contemporary neuroscientist named Bruce McEwen. And allostasis refers to imposing a change of the internal environment so that we can eventually cycle back and reestablish homeostasis. And I think these two terms, homeostasis, allostasis, they capture a sense of what the visceral motor system is all about. It's about trying to maintain the constancy of the internal environment and have the capacity to radically alter the state of the internal environment when the conditions demand for such action. So we'll see some examples of that as we go and talk about how these two aspects of visceral motor function are played out. Well, before we get into more detail about the anatomy and the function of our two principal divisions, our sympathetic and parasympathetic divisions, I just want to mention very briefly the enteric division. So the enteric division really refers to a network of neurons that are found in the lining of the gut. So here's an illustration that reveals just a small piece of representative tissue from the lining of our gastrointestinal system. For example, this could very well be the lining of the small intestine. And what I want you to notice are these networks of neuronal processes that run through particular layers of this tissue. So there are actually two networks. We call each network a plexus. So in close association with the muscular layers of the gastrointestinal system, especially the longitudinal muscles that are responsible for moving the contents of our gastrointestinal tract along through a pattern of muscle contraction that will propel in a wave-like fashion the contents of the gut along the GI tract. This plexus is called the myenteric plexus or Auerbach's plexus. So it's responsible for coordinating this rhythmical contraction of our longitudinal muscle. Well, there is a deeper plexus, and this plexus is found in close association with the mucosa that lines the gastrointestinal system. This is called the submucous plexus. And this submucous plexus, also known as Meissner's plexus, this is a network of neurons that coordinates the secretions of the various glands that are very important in mediating digestion within the gastrointestinal system. Now, these two networks of neurons, they can function in a semi-autonomous way. That is, if one were to have damage to the spinal cord or the nerves that supply this network, then this network can still function. However, the functions are not very well coordinated with respect to the needs of digestion. So there definitely is input to this enteric division of the visceral motor system from both sympathetic and parasympathetic sources. Nevertheless, there's a certain degree of autonomy that we recognize in the functions of this enteric division. Okay, well, that's all I'm going to say about the GI system. We certainly could say more. There's a rich degree of neurobiology in just about every way associated with the activities of neurons in these networks. But we'll turn our attention then to the sympathetic and parasympathetic divisions, and then the means by which the brain regulates these two sets of outflows from our visceral motor centers within the brainstem and the spinal cord. Okay, so I introduced two categories of molecules that help mediate the signaling that's required to grow circuitry in the developing nervous system. We've talked about cell-associated molecules. Now let's talk about the diffusible molecules. And they're essentially molecules that are secreted by target tissues and diffuse out into the extracellular fluids. These molecules can affect the growth and the survival of neurons and their individual processes, whether they be dendritic branches or axons. There are two categories of diffusible molecules based on their mechanisms of action and how they affect the development of the neuron and its processes. I'd like to talk first about tropic molecules. These are molecules that guide the growth of axons toward or away from the source of this particular molecule. So the directed growth is what we mean when we talk about tropism, and so this is a tropic molecule that directs the growth of an axon. They come essentially in two functional valences. There can be chemoattractant molecules or chemorepellent molecules. So I'd like to give you some examples of each. There are also trophic molecules. So while tropism refers to directed growth, trophism refers to nourishment. So these are molecules that provide some form of nourishment. Now, not nourishment in the form of glucose to be burnt up or ATP to be hydrolyzed and release energy from the chemical bonds, but nourishment in a way that promotes the survival and growth of the neurons and their processes. In the peripheral nervous system, these trophic molecules become really important in matching the axonal innervation with the target tissue that's present. And we'll also see the importance of trophic molecules for the development of appropriate circuits within the central nervous system as well. Now, one very important and very interesting aspect of trophic molecule production is that this is activity dependent. So this will provide the basis for competitive interactions in developing circuits. So a very interesting concept. We'll say more about that in our next tutorial. So let's consider some examples of our diffusible molecules. So again, the general mode of operation here is that these are molecules that are secreted by target tissues, and these molecules interact with receptors that are present on the surface of the growth cone. These diffusible molecules interacting with their receptors can have widespread effects on the neuron that transduces these signals. There can be local effects that can affect the growth patterns at the level of the growth cone and the individual axonal branch in question, but there might also be signals that go back to the cell body and affect gene transcription in the entire cell. So there are a variety of consequences of the interaction of these diffusible molecules with their receptors. So in this figure, we see illustration of several different kinds of diffusible molecules and their effect. We see some source of diffusible chemoattractant signals here. that begin to divert the growth of an axon towards that source of that chemoattractant. We also see contact-mediated chemoattractant signals that are important for helping to establish the growth of axons in parallel. And this is critical for the formation of axonal pathways, where there are many hundreds of thousands of axons typically growing together in parallel. Now, there are also chemorepellent signals that can be produced at some local source. And you'll see that as a growth cone begins to approach and explore, it may interact with one of these signals and then be diverted away to grow in the opposite direction from that source. There may also be contact-mediated chemorepulsant signals that help to make sure that the growth cones at the leading edge of the developing pathway begin to explore the entire environment, giving these growth cones an opportunity to seek out appropriate sources of chemoattractant and avoid the appropriate sources of chemorepellents. Now, also in this figure, we see the presence of trophic molecules that are expressed, and they don't necessarily direct the growth of an axon into the source of that trophic support. Rather, what they do is they validate that growth. So the growth is directed by the tropic molecules, but the survival and the ability of that axon in the neuron that gave rise to that axon to thrive is dependent in some measure on the acquisition of trophic support that comes through the interaction of these neurotrophin molecules and their receptors. We'll say more about that in just a moment. First, I'd like to give you some examples of some of the tropic molecules and then talk about one very important pathway that illustrates the operation of these molecules in development. So there are several representatives of these tropic molecules that we now know about. On the left, we see examples of the netrin and SLIT family of tropic molecules. Netrin is a soluble protein that is released and interacts with its receptors, DCC, and an associated protein called UNC5. So these interactions can result in the assembly of actin filaments, leading to the directed growth of that growth cone and its axon being elongated in its wake towards the source of netrin. Now, there's a related tropic molecule called SLIT that interacts with a receptor called ROBO, and SLIT antagonizes the activity of netrin. So netrin and SLIT together provide sort of a push-pull system or a go-stop kind of system that allows for the direction of growth. And this is particularly important at those forks in the road, as I'll show you in just a moment. Now, another diffusible tropic molecule illustrated here are the semaphorins. So semaphorins are cell-associated molecules that can be released and diffuse away from their source. And the semaphorins can interact with receptors, two of them illustrated here, plexin and neuropilin. These receptors can likewise interact with transduction machinery that can affect the assembly of cytoskeleton and the patterned growth of that growth cone. However, what the semaphorins tend to do is promote the disassembly of these neurofilaments, and that results in the retraction or the collapse of the filopodia and the lamellipodia. So the semaphorins are important signals that mediate the repulsive signals that are encountered as axons explore their environment. So let's consider one pathway, and indeed this is the pathway that I had in mind in our study question a little while ago. This pathway is the spinal thalamic tract. Now recall the second order axon of the spinal thalamic pathway. It has to do something which I find completely remarkable. This axon is grown out by a neuron in the dorsal horn of the spinal cord. And this happens throughout the entire length of the spinal cord. The axon will grow out very near its level of origin, and it will begin to grow in the anterior medial direction. So that axon encounters the midline of the spinal cord in the ventral white commissure, the anterior white commissure of the spinal cord. And it makes an important growth choice. It grows across that midline and enters the anterolateral white matter on the contralateral side of the spinal cord. So there is a decision to cross a midline here in the outgrowth of this second order axon. But there's also one more remarkable aspect to the growth pattern. Once across the midline and into the anterolateral part of the white matter, this axon now makes a hard turn in the superior direction and grows to the ventral posterior complex of the thalamus. So the implication is that there must be some chemoattractant signal that draws this axon towards the anterior midline of the spinal cord. And then once across the midline, there must be some signal that directs the growth away from that segment of origin and towards the superior axis. So we imagine there may be some combination of chemoattractant and chemorepellent molecules that achieve the necessary pattern of growth for this second order axon to establish the spinal thalamic tract. And indeed, what we imagine happening with the growth of this pathway is these second order axons, these commissural axons, are attracted towards the region of the floor plate. And they're attracted by the presence of netrin, which is secreted by this special region of the neuroepithelium called the floor plate. So these axons begin to grow in towards that floor plate. And then once they grow across the floor plate, now slit seems to be produced just lateral to the location of that floor plate. And slit now begins to negate the influence of netrin. And there's likely some other kind of signal, some other repulsive signal that is present here in the anterolateral part of the developing neural tube, perhaps a semaphorin, which will collapse the growth cone from growing in this direction and rather promote growth away from the source of that chemorepellent. There very likely is also some kind of chemoattractant that is in some concentration gradient drawing this axon towards the superior aspect of the developing hindbrain, even into the forebrain, to reach its ultimate target in the diencephalon. So this is a wonderful example of how the presence of chemoattractant and chemorepellent molecules can mediate what are really some spectacular patterns of growth that we've learned about in our survey of the structure of the sensory and motor systems. I'll refer you to a beautiful box in chapter 23 that talks about another famous fork in the road. That would be the optic chiasm, where similar functions seem to direct the nasal retinal axons across the optic chiasm while repelling the temporal retinal axons, which remain on the ipsilateral side of the brain, progressing through the ipsilateral optic tract. So turn to the box there in chapter 23 to learn more about that particular pathway. Okay, well, now let's turn our attention to the parasympathetic division of the visceral motor system. And we see that illustrated here on the left-hand side of figure 20.1. So the parasympathetic division, likewise, is a two-neuron connection between the central nervous system and the viscera. However, the first neuron tends to project a much longer distance than did the first neuron or the preganglionic neuron on the sympathetic side. And the second important difference about our preganglionic neurons is that in the parasympathetic division, those preganglionic neurons are found in a different location. They're not found in the thoracic cord, they're found in the brainstem, and then in the inferior end of the spinal cord, in the sacral segments of the spinal cord. So the preganglionics are found in different locations in the sympathetic and parasympathetic side, and their axons tend to go quite a different length, with the parasympathetic preganglionic axons being distributed typically to the end organ itself, where the ganglia that receive the synaptic connections are embedded in the end organ itself. Consequently, the ganglionic neuron in this system extends only a short distance to reach the effector tissue itself. So this illustration is not quite anatomically accurate in that respect. These ganglia are often embedded in the tissue itself. So the locations here are meant to be more illustrative of this relationship between pre- and post-ganglionic neurons rather than describing their actual anatomical location of the ganglia that serve the viscera. Okay, well, with that said, let me just comment about the neurotransmitters since we're here on this slide, and then we'll move on and see in more detail the locations of these preganglionic neurons. So the neurotransmitter of the preganglionic neuron, like the sympathetic division, is acetylcholine. So these preganglionic parasympathetic axons that supply these targets emanating from the brainstem and from the sacral spinal cord are going to release acetylcholine in their ganglia. Well, the postganglionic neurotransmitter of the parasympathetic division is also acetylcholine. So acetylcholine is a prominent neurotransmitter on both sides of this system. Now, I should mention on the sympathetic side, there is one place where we know that acetylcholine is the ganglionic neurotransmitter, and that's right here in our sweat glands. So the sympathetic division is what activates our sweat glands, and it does so via the mechanisms of action of the neurotransmitter acetylcholine. But otherwise, we associate acetylcholine with the parasympathetic ganglionic neurons, and we associate norepinephrine with the ganglionic neurons on the sympathetic side. Okay, well, let's now have a look at the anatomy of these preganglionic neurons of the parasympathetic division of the visceral motor system. Looking first at the brainstem. So these neurons are found in 4 pairs of nuclei of the brainstem. And beginning in the midbrain, we have the Edinger-Westphal nucleus. Now, I hope you will recall that this is the small parasympathetic nucleus that's associated with the 3rd cranial nerve. This Edinger-Westphal nucleus is found just dorsal and a little bit lateral to the somatic motor neurons of the ocular motor complex. So we can find that in Sylvius, and I'd like to point that out in just a moment. But for now, just know that the Edinger-Westphal nucleus is a parasympathetic nucleus of the midbrain. The remaining parasympathetic nuclei of the brainstem are found in the caudal pons and in the medulla. So in the caudal pons, we have a set of nuclei that we call the salivatory nuclei. So these nuclei provide for parasympathetic outflow to our tear glands and our saliva glands. Now, just at the junction of the caudal pons and then down into the upper part of the medulla, we have our dorsal motor nucleus of vagus and the nucleus ambiguus. Now, the dorsal motor nucleus of vagus is a primary source of axons that supply visceral targets via the vagus nerve. One additional contribution to that vagus nerve that is very important in governing the activity of the heart comes from a set of cells that are anatomically associated with the nucleus ambiguus. So the nucleus ambiguus really has at least 2 distinct populations of neurons. There are the branchial motor neurons that supply the muscles of the larynx and the pharynx, and then there are these parasympathetic preganglionic neurons that are involved in slowing the heart. Okay, so these are the 4 major sources of parasympathetic preganglionic neurons in the brainstem, and here they are in cross sections. So here's our Edinger-Westphal nucleus up in the midbrain. Somewhere in this location, we don't need to attempt to find it precisely, are the salivatory nuclei. As my colleague Nell Kant, who helped me write these tutorial notes for you, often notes that there are very few people in the world who know where to find these salivatory nuclei in the caudal pons in the upper medulla. Well, a little bit more prominently and a little bit easier to find is the dorsal motor nucleus of vagus and the nucleus ambiguus, which are found in the upper part of the medulla. So here we are in Sylvius, and let's just find these structures. So if we look at the midbrain section, what you'll see if we follow back the nerve roots of the oculomotor nerve is our oculomotor nucleus. So this is the source of the somatic motor outflow that governs those striated muscles that we have in the orbit, and a levator muscle that opens our eyelids. But just on the dorsal aspect of this oculomotor complex is where we find our Edinger-Westphal nucleus. So again, this nucleus gives rise to parasympathetic preganglionic axons that have their cell bodies here, and then they grow their axon out through the third nerve to constrict the iris in the eye. All right, so next let's jump into the caudal part of the pons. And somewhere in this tegmentum, in the core of the brainstem, is where we would find our salivatory nuclei. So we don't identify them in Sylvius. I don't happen to be among the small number of people in the world that know precisely where to look for these neurons. I suspect I could find them if we were looking at a Nissl stain, but in a myelin stain, there just aren't enough cells to produce a distinct cluster of gray matter that we could recognize in a myelin stain here. So I won't attempt to point out where those salivatory nuclei are. However, if we go into the upper part of the medulla, maybe just go down one more section, I certainly can highlight the location of the dorsal motor nucleus of vagus. So this is a major parasympathetic preganglionic outflow that supplies for much of the innervation of the vagus nerve throughout the viscera of the thorax and the abdomen. Now, in a more anterior position in the tegmentum, we find the location of the nucleus ambiguus. And so with respect to visceral motor control, this is where we find our cardio-inhibitory parasympathetic preganglionic neurons. Well, that's it for the parasympathetic preganglionics of the brainstem. To find the rest, we need to move into the sacral segments of the spinal cord. So let's do so and find our way down to the most inferior section in our brainstem spinal cord atlas. And here we are in the sacral level of the spinal cord. I know that because there's a very large ventral horn and not much white matter associated with the spinal cord. And if we look into the dorsal column region, we find that there is only one division of the dorsal column, a gracile tract, no cuneate tract. So hopefully those cues are now beginning to register with you as you look at cross sections at different levels of the spinal cord. Okay, so we're looking for our preganglionic parasympathetic neurons, and we find them in a location that's somewhat similar to where we found a lateral horn of the thoracic cord. But in the sacral cord, we really don't have a distinct lateral horn. Rather, we just have a fairly smooth outline of the dorsal horn and the intermediate gray between the dorsal and the ventral horn, and then of course a very large bulging out ventral horn. So we don't have a lateral horn, but we do have a column of cells that we can recognize as our sacral autonomic nuclei, or our preganglionic parasympathetic nuclei. So this is where they're found. Okay, so let's return to our slides and consider this arrangement in a more illustrative configuration. So here again, in a view of the intermediate gray matter of the sacral cord, we find the location of these neurons. Now, as I mentioned in broader view, what these neurons actually do is they exit the spinal cord through the ventral roots. They bypass our paravertebral sympathetic trunk, and rather they make their way to ganglia that are associated with the viscera. And that's where we find our synapse with our postganglionic neuron. And the postganglionic neuron then supplies the visceral tissue, whatever that target organ happens to be. Now, before we move on and consider more central aspects of visceral motor integration, I want us to just make a couple of points of emphasis here. Now, I want you to understand that there's always coordination of activity in our sympathetic outflow and our parasympathetic outflow. Yes, they do represent rather extreme ends of a continuum of function, but that function is a continuum, and there is tone in the outflow of each division all of the time. So while sometimes it's helpful to emphasize, for example, that our sympathetic division is about fight or flight, or the mobilization of resources for action, I want you to understand that that's not the only circumstance in which there is outflow from our sympathetic division. And likewise, for our parasympathetic division, sometimes we consider this to be a division that is restoring resources in times of resting and digesting, and it's perfectly fine to use those kinds of monikers as a way of understanding the principal actions of these divisions. But what I really want you to get is the idea that there is a continuum of action here, where there is tone in each side of this visceral motor outflow. And the couple of points that I want you to take away from this are that the functions are complementary, but they're coordinated. There's always some tonic activity in both divisions of the visceral motor outflow. And while it's possible to increase activation in a wholesale fashion across multiple visceral organs, that is not necessarily how these functions are coordinated. Rather, there can be local coordination of sympathetic and parasympathetic outflow that can serve to coordinate the activity of just one organ system without necessarily engaging the entire visceral effector system in an all or none fashion. Well, one important context in which this ongoing coordination of parasympathetic and sympathetic activity is played out at the organ-specific level reflects the operation of reflex activity. So I want us to now begin to move into higher levels of processing in the central nervous system. But before we get there, I want you to appreciate the fact that there is a rich source of sensory information that is coming into the central nervous system, and this information is integrated at various levels. At one level of input, there is integration in an important nucleus of the brainstem that I'll talk about in a little bit more detail in just a moment, called the nucleus of the solitary tract. There's also integration in the spinal cord for the viscera of the abdomen, especially. And then there is outflow from this network that's integrating these sensory signals to preganglionic neurons that then can engage a visceral motor response. So this kind of reflex activity is not unlike our segmental reflexes that organize the output of our alpha motor neurons, as we've talked about when we discussed the myotactic reflex, for example. So there's sensory signals that are integrated at a very local level, and that can influence the output of our preganglionic neurons, both parasympathetic and sympathetic. In this way, the activity in a visceral effector system can be regulated in a moment-to-moment basis with adjustments in the tone of our sympathetic or parasympathetic outflow, depending upon the organ system in question. Well, I want to say a little bit more about the sources of this visceral sensory input. So this visceral sensory input essentially comes through two cranial nerves and through collaterals of our anterolateral pathway. So in this illustration, we see those two cranial nerves. They are the glossopharyngeal nerve, cranial nerve 9, and the vagus nerve, cranial nerve 10. So the sensory information comes via the neurons that are associated with the ganglia of these two cranial nerves, and their peripheral process is very much like our somatic sensory elements that we've already discussed, mechanosensory axons, nociceptive axons, free nerve endings. But in the case of our visceral motor afferents, there are also specialized tissues in certain regions of the body that are sensitive to the chemistry of the blood that's flowing past these structures, or the chemistry of the extracellular fluids. And so there are chemoreceptors that are sensitive to changes in pH, in carbon dioxide, in oxygen tensions, for example. So there are mechanosensory afferents, there are nociceptive afferents, and there are chemosensory afferents, all supplying information that is fed into the central nervous system. So, as I mentioned, an important integrator of this incoming information is this nucleus of the solitary tract. And we've already talked about this nucleus when we talked about our gustatory system. As you'll recall, this nucleus really has two principal divisions to it. There's a rostral division, which is receiving this incoming special visceral sensory information about taste. And then there's a caudal division, and that's the part that I want to focus on here. It's this caudal division that's receiving this input from these cranial nerves and from collaterals of our anterolateral system. So this nucleus of the solitary tract then is really a key center that integrates incoming visceral sensory signals. So just to give you a view in a brainstem cross-section, this nucleus is really quite distinct. It's found in the upper part of the medulla, and we recognize it as something that looks sort of like a bullseye. There is a dark myelinated spot right in the middle of the nucleus. So that spot, that is actually the axons of these afferents that are entering this nucleus. And it's dark because these afferents are well myelinated. And the gray matter of the nucleus of the solitary tract surrounds this tract. So hence you can understand why this is called the solitary tract, because it's sort of sitting there by itself in isolation. And it's isolated then by the gray matter of the nucleus that surrounds this solitary tract. Well, that's where you would find this nucleus, the upper part of the medulla, the dorsal aspect of the tegmentum. Now, I want to make just one other comment about visceral sensory afferents, and it's reflecting some fairly recent discoveries about the way that visceral pain is processed in the central nervous system. There is now a well-understood pathway that runs through the medial part of the dorsal columns of the spinal cord that conveys visceral pain information. Now, this is in addition to the pathways that run through our anterolateral system, so I don't want you to hear me saying that this conflicts with what I just said about the anterolateral afferent supplying the nucleus of the solitary tract. Rather, this is an additional pathway that was discovered in the late 80s and the early part of the 90s. And what we think is happening here is a pathway that connects especially the lower GI system from the first order afferent, which would be a nociceptive element in the appropriate spinal root, to a set of second order neurons that are found near the central canal of the spinal cord. And these neurons send an axon not to the anterolateral system as we might have anticipated, but rather up along the medial edge of the dorsal columns. And there they synapse with neurons in the dorsal column nuclei that grow an axon that appears to enter the medial lemniscal pathway and make synapses on neurons in the ventral posterior complex that project not up to the postcentral gyrus, but rather into the insular cortex, where representation of visceral sensory information seems to be elaborated at the level of the cerebral cortex. Well, this pathway is worth mentioning because it highlights an important clinical phenomenon that I should mention, and that is referred pain. Now, referred pain is often considered a phenomenon involving a crosstalk between dorsal root ganglion neurons that supply, let's say, skin tissue, and dorsal root ganglion neurons that might supply visceral tissue. So here I'll just indicate that this is maybe the lumen of the gut, and there's some kind of a visceral sensory axon innervating that tissue. Well, the central processes of these neurons might very well converge upon the same cell in the dorsal horn of the spinal cord. So here's the spinal cord. Let's just say this is the dorsal horn, and somewhere in here is a second order neuron of the anterolateral pathway. So one might imagine that the pain associated with this pathological process in the viscera might actually get referred to the more peripheral structure. And the fact that these patterns are so predictable suggests that they actually do align with the organization of the body map in the dorsal horn of the spinal cord according to the segments of the spinal nerves. So illustrated here, for example, are visceral pain patterns associated with the esophagus, with the heart, and with the left ureter. So this dorsal horn crosstalk hypothesis provides a reasonable framework for explaining these referred pain patterns. Well, it has occurred to me, certainly, and others, that this newly discovered dorsal column visceral pain pathway provides yet an additional possible explanation of referred pain. Consider the gracile nucleus, for example. It's integrating inputs from the lower part of the body, from mechanosensory afferents that are more peripheral in their distribution, in the cutaneous surfaces, in the deeper tissues of the limbs, for example. But in experimental studies using non-human primates, for example, we know that the very same gracile nucleus neurons may also respond to distension of the bowel. So they seem to be receiving input from visceral nociceptors in addition to their mechanosensory afferents concerning the lower extremity. So this might be yet another means by which some kind of crosstalk might occur at the level of innervation of a distinct gray matter structure. So just keep in mind, referred pain doesn't necessarily have to happen via interactions in the dorsal horn of the spinal cord. It actually might happen at multiple levels of our somatic sensory pathways. At least I think this gracile nucleus is a good candidate where some visceral pain might be referred to a more peripheral target. Well, let's get back to thinking more broadly then about the central integration of visceral sensory signals as the output of the visceral motor system is organized and ultimately coordinated. Okay, what I'd like to do now is introduce this concept of central integration of these visceral sensory signals so that the motor output can be coordinated by higher brain centers in the medial and ventral aspects of the forebrain. So I want to show you really an expanded, or maybe you might think of this as an exploded version of this illustration that unpacks exactly what these structures are and how they relate to the influx of sensory signals and then the output of our preganglionic neurons. So this is the last slide that I want to show you in this part of our tutorial, and trust me, it looks actually much worse than it really is. Let me give you a sense of the organization of this chart. What we have on the right-hand side of this chart is basically the ascending sensory information that is derived from our visceral sensory elements. Many of these things we've already discussed. So our end organs are giving rise to first-order afferents that are arising through cranial nerves 9 and 10 and the axons of our anterolateral system. And the nucleus of the solitary tract then becomes a very important integrator of this information. We've already talked about how the nucleus of the solitary tract gives rise to projections that impact our interneurons that organize the preganglionic neurons of the sympathetic and parasympathetic divisions in the brainstem and the spinal cord, and this thereby mediates local reflex activity that can alter the activity of our end organs. So what I want to introduce to you is the idea of a central autonomic network that is processing these ascending sensory signals and coordinating the output of our visceromotor effector systems through a broad distributed network of elements in the ventral medial parts of the forebrain, including some diencephalic regions, including the hypothalamus, very important structure that I'll talk about next, as well as parts of the brainstem. So from the nucleus of the solitary tract, inputs are passed into the reticular formation of the brainstem. They're also distributed directly to the hypothalamus and then indirectly to another nucleus of the brainstem called the parabrachial nucleus. This is found very near the superior cerebellar peduncle as it attaches to the upper part of the pons. Well, this parabrachial nucleus then sends input to the medial parts of the thalamus, to the hypothalamus, and to the amygdala. And once this information has access to the thalamus and the amygdala, now it can be distributed more broadly to the ventral and medial aspects of the prefrontal cortex. The thalamus and the amygdala have connections specifically with the insular cortex, and the insular cortex in turn has interconnections with the rest of the orbital and medial sector of the prefrontal cortex, which becomes very important in integrating the sensory signals that are being processed and impacting the motor output that eventually feeds its way down towards our brainstem and spinal cord networks of neurons. Well, let me just pause for a moment and highlight the importance of this insular cortex. We can think of this insular cortex as being the sensory cortex for the viscera. So one aspect of this insular cortex that I think is really interesting is its capacity to integrate these visceral signals and then use that information as a way of informing other kinds of networks that mediate different dimensions of cognition. So here's an example. Let me ask you a question. How do you feel right now? Well, if you're like me, you may respond based on information that is derived more so from the viscera than, let's say, from our cutaneous surfaces. Now, of course, you may have a condition by which your cutaneous surfaces or your musculoskeletal systems really drive the answer to that question, how do you feel? But I think for many of us, that response, whether we think of it in these terms or not, is really derived from our visceral senses. And that is a signal that is processed here in this insular cortex. So I think this insular cortex is very important for this notion of feeling. So how do you feel today? Well, let your insular cortex inform the rest of your cognitive networks as you consider your reply to that question. All right, well, let's move from the right side of this figure then to the left side. So as the insula has interactions with the rest of the medial and orbital aspects of the prefrontal cortex, we begin to coordinate and organize our plan of action, our governance of our visceromotor output. And this happens through multiple relays, some of which run through the amygdala and then through the hypothalamus. Others enter the amygdala and hypothalamic systems and then have more or less direct access to brainstem networks that organize our preganglionic, sympathetic and parasympathetic neurons. Well, eventually from that level then, of course, we have our motor outflow to our ganglionic neurons that can alter the activity of our end organs. So we might think of this medial prefrontal cortex then as the principal coordinator of outflow that is going to alter either our homeostatic state or prepare our body for action, that is for allostasis, to change the state of the body in order to once again restore conditions of homeostasis. Well, an important mediator then of activity from the level of the cortex to these brainstem effector systems in the reticular formation and then the preganglionics themselves is this part of the ventral diencephalon called the hypothalamus. The amygdala is important too, but we'll talk about that in a later session in the final unit of the course. I'd like to take a few minutes speaking with you about the hypothalamus and highlight for you its key role in integrating visceral sensory signals as well as command signals and contextual signals derived from the level of the cerebral cortex. And then the outflow from the hypothalamus is largely directed at our visceral motor effector systems, as we'll see in the next part of this tutorial. So if you'll hang with me, we'll move from this rather lengthy first part of the visceral motor tutorial, and I want to spend some time with you talking about the hypothalamus. But before we get there, consider this study question, and I think you'll be ready to move on after that. So I'll see you in just a minute, and we will pick up talking about the hypothalamus. Welcome back to the next part of our tutorial on the visceral motor system. In this session, I want to focus on the hypothalamus. So again, our discussion relates to the complexity of the brain and some of the circuitry of the brain that is genetically determined, and it provides the foundation for the operations of the elements of the nervous system that are the focus of this session. And for us, it's going to be the circuitry of the hypothalamus. So my learning objective for you in this session is that I want you to be able to describe the organization of the hypothalamus and identify several functions associated with key hypothalamic nuclei. Well, we concluded the first part of our visceral motor tutorial by focusing in on the central autonomic network. And we talked about the ascending aspects of sensory signals that are derived from the viscera and how they are integrated within this central autonomic network. And the outflow back down to our visceral motor end organs is coordinated in a large measure by this key structure in the ventral part of the diencephalon called the hypothalamus. So I think it will be well worth your time to just think with me for a few minutes about the hypothalamus, its structure, and then the myriad functions associated with the circuits that are localized to different structures that are part of the hypothalamus. Okay, so with that as our context, let's look at the anatomy of the hypothalamus. So I'll just remind you, with this mid-sagittal view of the human brain, that the hypothalamus is found in the ventral aspect of the diencephalon and in a somewhat anterior position. Now, I've drawn a triangle here because this is roughly the region where we find the hypothalamus. So this triangle is essentially the third ventricle, and the hypothalamus forms the walls of the third ventricle and obviously extends from the ventricular surface into the depth of the brain and this ventral part of the diencephalon. So this triangle is really formed by three structures that I think you can see quite plainly here in this image. So one structure is found right here, and this is a white matter structure that crosses the midline. Now, it's not the corpus callosum. The corpus callosum is up here. It is another commissure. So perhaps you can think of what that structure is. I'll give you just a moment. And everyone shout out together. It's the anterior commissure right here. So that anterior commissure is a bit of a visual landmark that defines one point of this triangle that can help bound your recognition of the hypothalamus. Okay, so the low point of the triangle is right here. And again, does anyone know what this is? This is a structure associated with the visual pathway connecting the retina to the diencephalon and the pretectal region of the upper part of the brainstem. This is actually the optic chiasm. Okay, so hopefully you can recognize that. So those are 2 points of the triangle. The third point is actually a part of the hypothalamus itself, and that is this medial ball-like structure that sits very close to the midline. There's a pair of them, one on either side. And again, who knows what this is? This is one of the two mammillary bodies. Okay, so these are the three visual landmarks that form this triangular region that allows us to recognize the location of the hypothalamus. Well, this is the gross view of the hypothalamus. A more detailed anatomist view of the hypothalamus would really be quite different. So if we were to look now at this triangular region in some detail, here again is our triangle connecting our anterior commissure, our optic chiasm, and our mammillary body. What we find is that the hypothalamus is not just one uniform structure, but it really is a region that consists of many different nuclear divisions. In this respect, the hypothalamus is quite like the thalamus, where in the thalamus, as hopefully you're now quite comfortable with this idea, there are a number of discrete nuclei, discrete clusters of cells that have distinct input and output connections. And the information that's conveyed via those inputs and outputs helps to define the functional distinctions of each of those divisions. Well, the very same concept applies to the hypothalamus. So what we find in this figure is a rendering of just a subset of the nuclear divisions of the hypothalamus that might be recognized. So the color code just organizes these nuclei in a rostral to caudal or an anterior to posterior progression, but there's also a medial to lateral progression of nuclei that I want to highlight for you in just a moment. Well, the big picture here is that the hypothalamus is comprised of a number of discrete nuclei that maintain distinct patterns of input and output connections. And many of these nuclei are associated with very particular functions. And I'd like to highlight a few of them for you in just a little bit. But before we get there, what I'd like to be able to do is give you a bit of a broader picture of what the hypothalamus and its many nuclei are involved in. I'd like to give you a sense of the general mechanism of hypothalamic function in each of these circuits, and then we'll talk through some particular examples that will give you a sense of what kinds of functions might be associated with hypothalamic nuclei. Okay, well, thinking very broadly, I would assert that the hypothalamus regulates 5 basic functions. It regulates blood pressure and electrolyte balance. And it does so by modifying a variety of behaviors and physiological functions that can impact blood pressure and electrolyte balance, such as seeking out fluids, drinking, generating an appetite for salts, adjusting the osmolarity of our blood, and adjusting our vasomotor tone, which, as you know, has an impact on our blood pressure. The hypothalamus can regulate body temperature, and it can do so again through various physiological and behavioral means. The hypothalamus has governance over thermogenesis in the physiological systems of our body. The hypothalamus can also motivate behaviors that result in seeking warmer clothing or warmer environments, or the removal of clothing and the seeking of cooler environments. The hypothalamus can regulate energy metabolism by coordinating our feeding behaviors. This is a huge topic. I wish we had more time in medical neuroscience to really unpack the role of the hypothalamus in the feeding system. But perhaps in future editions of the course we'll try to create some additional time for that. Well, the hypothalamus helps to regulate the intake of food and its digestion, and it also helps to establish a sense of satiety when our intake of food should cease. And as you might imagine, there are a variety of systems that can become dysregulated and can result perhaps in a variety of eating disorders, including obesity, which is a huge problem in many parts of the world, and including here in the United States. So we definitely need to direct additional attention in understanding the governance of our feeding system and the various dysfunctions or dysregulations of this part of the brain. Well, the hypothalamus is also involved in adjusting metabolic rate, which also obviously has a major impact on energy metabolism in the body. The hypothalamus has really a fascinating role to play in governing reproduction, and this involves a variety of different aspects of reproductive activity. It involves the physical behaviors that are associated with sex and mating activities. It involves a sense of sexual identity, and sexual orientation can be influenced by hypothalamic circuits. There are hypothalamic circuits that operate in females to coordinate the various physiological changes that are necessary to support pregnancy, and then in the postpartum period, lactation and the nourishment of the infant. And then lastly, the hypothalamus is very important for coordinating the responses that support what we call allostasis, that is, changing the internal environment of the body in order to respond to emergency conditions. So the hypothalamus is involved in coordinating the distribution of blood flow to where it's needed most, given the contingency that we're faced with. It is involved with coordinating the release of hormones, governing the neuroendocrine relation, and in this case of emergency responses, increasing the release of stress hormones that help to mobilize the body for action. And the hypothalamus even has interactions with our immune system that likewise are important for mediating how we respond to emergency conditions. Well, this list could be longer than 5 items, as I'm sure you can imagine, and perhaps we could kick to a discussion forum, a richer discussion of the various dimensions of hypothalamic coordination of homeostasis and allostasis, but I think I'll leave it here for now. And now let's talk about the general mechanisms by which the hypothalamus can control these 5 basic functions. Well, I see the hypothalamus as sitting in a key position in the nervous system anatomically and physiologically. It's getting sensory inputs from visceral and somatic sensory pathways. It is sensitive to chemosensory signals. In some parts of the hypothalamus, directly sensitive to the chemistry of the blood and the extracellular fluids. In other parts, receiving ascending sensory information from the nucleus of the solitary tract and its relays into the hypothalamus regarding the chemical environment of the body. The hypothalamus is also sensitive to the signaling derived from humoral factors that are circulating in the blood. Well, all of this sensory information is integrated with contextual information that's derived from the telencephalon, largely the orbital and medial parts of the prefrontal cortex. Other parts of the cerebral cortex project to the hypothalamus as well, as does the amygdala and the hippocampal formation in the medial parts of the temporal lobe. So the hypothalamus is integrating context, it's integrating sensory signals, and it's giving rise to output signals that are coordinating our visceral motor, our somatic motor, and our neuroendocrine systems. And the result is the production of appropriate behavioral responses that reflect the coordinated activity of all of these different dimensions of movement to produce behavior. One important concept in hypothalamic control that we're still trying to understand is this notion of biological set points. So this integration of context and sensory signal is referenced to some measure of homeostatic set point. Now, we don't really understand this concept as it's played out in neurobiological terms for very many of the systems that are coordinated in the hypothalamus, but we suspect it's a common mechanism. And the implementation of this mechanism undoubtedly gets us deep into the molecular biology of this circuitry and how molecular mechanisms are mediating these set points, and how signaling within the neuron and perhaps even within the nucleus is producing patterns of gene expression in space, in particular circuits, and in time, in different conditions that demand different sorts of responses. Well, we'll learn a little bit more about this when we talk about the regulation of sleep and wakefulness. There are circuits in the hypothalamus for which the molecular set points are reasonably well understood, so I'm going to unpack a little bit of that in a later session. But for now, I'll just broadly emphasize this general mechanism of hypothalamic function of integrating context and sensory signals and referencing against a set point. and with deviation from that set point, we have implementation of some kind of behavioral change. Okay, well, let's now look in a little bit more particular detail at different nuclei of the hypothalamus. And I want to give you a sense of what is the diversity of function that is attributable to different nuclei within this collection that we call the hypothalamus. Now, I don't intend for you to know all of this detail. I really don't. I give some of this to you in the tutorial notes, mainly as a way for you to just spend a few minutes thinking together with me about the hypothalamus and the diversity of systems that it impacts. There is one, however, that I want to highlight for you because we're going to come back to this in a later session. Okay, I mentioned one can look at the hypothalamus in different axes, from front to back, from anterior to posterior, from medial to lateral. What I want to do is just highlight a few nuclei that catch my attention, at least, going from the medial to lateral axis of the hypothalamus. And let's begin right along the walls of the third ventricle. There's a very interesting pair of nuclei that we find right along the wall of the third ventricle and just inferior to it. These nuclei are found in what we might call the periventricular zone, right around the third ventricle. There's a nucleus called the periventricular nucleus, and there's another one called the arcuate nucleus. And these nuclei are intimately related to the production of hormones in the anterior part of the pituitary gland. This is the part that we call the adenohypophysis. So the anterior pituitary receives hormones that are produced by cells that sit in this periventricular region of the medial hypothalamus. And those hormones enter the portal circulation, where they are then transported via this blood supply to the anterior part of the pituitary. These hormones, we call them releasing factors, then have an impact on the neurosecretory cells of the anterior pituitary. And these cells then produce a hormone that enters the general circulation and can impact physiology throughout the entire body. So right along the medial border of the hypothalamus is where we find those neurons that govern the output of the anterior part of the pituitary. Well, moving on just a bit more lateral, in the color code here, these would be these darker solid shaded regions in here. We have a variety of interesting nuclei that do all kinds of different functions. Let me just highlight a few of them. There is a set of nuclei called the paraventricular nucleus and the supraoptic nucleus. So here's the supraoptic nucleus. It's called supraoptic because it sits just above the optic chiasm, and in a slightly more dorsal position is this paraventricular nucleus. These nuclei are two of many that we find in this more medial zone of the hypothalamus. I mentioned the paraventricular and supraoptic nuclei because these are the nuclei that have the neurons that grow their axons through the infundibulum, that is the stalk of the pituitary, and release hormones directly into the general circulation in the neurohypophysis, or the posterior part of the pituitary gland. Okay, so the periventricular zone is where we find our neurons that are engaging the anterior part of the pituitary, and then in this more medial zone, that's where we find this supraoptic and paraventricular nucleus that are releasing hormones into the posterior pituitary. Well, there are other neurons in this paraventricular nucleus in particular that are not releasing hormones in the pituitary, rather they're growing axons that descend into the brainstem and even into the spinal cord. And these paraventricular neurons that grow these long axons are involved in coordinating the output of preganglionic neurons in the parasympathetic and in the sympathetic divisions of our visceromotor outflow. So paraventricular nucleus is a very important structure in regulating various dimensions of our behavioral response. Okay, I'd like to highlight a few more nuclei here in this medial zone. Generally speaking, this medial and lateral preoptic area here, and I'll highlight the medial preoptic region. This is a really fascinating part of the anterior hypothalamus. This is a part of the hypothalamus that is involved in various aspects of coordinating reproduction and sexual behavior. So this is a part of the hypothalamus that we think is especially involved in defining our sexuality and motivating sexual activities. And here there are circuits that are coordinating various aspects of mating behavior, and in women, various dimensions of pregnancy and postpartum behavior. So this preoptic area has connections with important parts of our limbic forebrain that are involved with emotion. As you might expect, our emotions are important sources of input to circuits that are mediating sexual behavior and sexual activities. This medial preoptic area is also involved in governing the release of urine in the process of micturition. We'll have more to say about that in the final part of this tutorial, but just keep that in mind. The medial preoptic area is also involved in governing bladder function. Other structures in this medial zone worth mentioning include the suprachiasmatic nucleus. So this is a small nucleus that sits just above the chiasm here, just a little bit medial and anterior to where I highlighted the supraoptic nucleus. So this is the suprachiasmatic nucleus. Hopefully that's easy to remember. And I do want you to remember this nucleus because this is a nucleus that we'll come back to when we talk about the regulation of sleep. The suprachiasmatic nucleus is the master clock of the human body. This is the place where we have neurons that generate a circadian rhythm. That is a rhythm that runs roughly 24 hours, not precisely 24 hours. We'll mention that in a later tutorial. But it allows us to establish a daily rhythm for behavior, for the release of hormones that can affect our body's physiology and our behavior. So very important part of the hypothalamus, and it's that part that I mentioned when we discussed the visual pathways that receives input from a special set of photosensitive ganglion cells in the retina. So by this connection with the retina, this suprachiasmatic nucleus can be entrained to our natural cycles of light and darkness, of daytime and nighttime. There's another set of medial zone nuclei that I would mention. The dorsal medial nucleus and also the ventral medial nucleus. These two nuclei seem to be involved in some other dimensions of reproductive behavior and even parenting behavior, at least based on animal studies. One might expect that. These nuclei seem to contain circuits that are involved in the feeding system, helping to motivate the consumption of food, as well as mediating signals that are related to satiety. So very important networks here motivating the ingestions of foods and possibly also particular appetites for things like salts or particular food substances that we might happen to be in deficit of. There are other circuits here that are also involved with water balance and thermoregulation. Well, lastly, I would highlight this more lateral part of the hypothalamus, indicated by this stippled region here. So there are nuclei out here more laterally that are involved in really a variety of functions that pertain to attention and arousal. Now, these aren't the only neurons in the brain that are important for those kinds of functions, but these parts of the hypothalamus seem to have broad connections, especially with the cerebral cortex, and they seem to be in a position to modulate the way the cortex responds to other kinds of inputs. So in that sense, the lateral hypothalamus seems to have a role to play in modulating levels of vigilance in the brain in a manner that might be similar to signals that are motivated by the amygdala, as well as some of the biogenic amine systems in the brainstem. Well, as I said when we got into this slide, there are way too many details that are beyond the scope of this course to expect you to take in this brief tutorial. Rather, what I want you to do is at least remember this important suprachiasmatic nucleus, because we're going to come back to that. but even more so, I hope you get just an impression that there are a variety of physiological functions that can be absolutely central to motivated human behavior that are coordinated at the level of the hypothalamus. Well, now that you've had a bit of a sense of some of the functions associated with nuclei of the hypothalamus, I want to conclude this session by talking about a particular clinical condition called Horner syndrome. Now, Horner syndrome is relatively common. We might see it in a variety of patients for different reasons, and it is a presentation of signs and symptoms that are typically associated with the face. Now, Horner syndrome could be bilateral, but it more commonly is unilateral, and it's characterized by a set of signs that can be best appreciated clinically by focusing on the region of the eyes. So what we find most significantly is miosis, which is a constriction of the pupil. Now, what you're looking for when you examine the diameters of the pupils is not so much an absolute size, but an asymmetry. In the case of Horner syndrome, what we see is the affected iris has a smaller pupil than the normal iris. Well, it might be somewhat difficult to appreciate what is normal, but I think you can see that the dimension of the pupil is smaller in this patient's right eye compared to the left eye. So this is miosis. It's evidence of an abnormal constriction of the pupillary muscle. Well, in addition to the constriction of the pupil, we also see a drooping of the eyelid associated with Horner syndrome. So this might be a subtle sign, but it's one that you should look for nevertheless. And one might also get the impression that the eyeball itself, the globe, is somewhat sunken into the orbit. There are some other signs and symptoms associated with Horner syndrome that may be a little bit more difficult to appreciate clinically, but perhaps through patient interview you can confirm their presence or absence. One sign you may be able to detect if you were to put your fingertips on the cheeks of your patient, you may discover that the side with the constricted pupil actually feels a little bit warmer to the touch than the opposite side. Now, if you were to ask the patient about their sweat response to hot environments or to exercise, you may discover that the side with the smaller pupil doesn't actually seem to sweat as much as the opposite side of the face. Well, can you think of what all of these signs might have in common? Do you remember what's responsible for the dilation of the pupil? What about the ability to keep our eyelids open throughout the day? And what system supplies innervation to the sweat glands? Or what do you think an increase in the temperature of the skin might tell you about the tone of the vasculature that's supplying these cutaneous structures of the face? Well, hopefully I'm leading you along the path that allows you to understand that what all of these systems have in common is sympathetic control. Well, Horner syndrome can result from an interruption in the pathway that links the hypothalamus up here in the ventral and anterior part of the diencephalon with our ganglionic neurons, which, with respect to the governance of these structures in the face, are localized to the superior part of that sympathetic trunk. Specifically, the pupillary dilator muscle is supplied by ganglionic neurons that sit in the superior cervical ganglion and grow an axon through the carotid plexus to reach the orbit where there's innervation of this dilator muscle of the iris. So just given the broad distribution of this system from hypothalamus to face or to iris, you can imagine that one can have a lesion or some other kind of disease process that would affect this pathway, either within the nervous system or outside of the nervous system, and produce Horner syndrome. And indeed, one might have damage to the neck that's outside of the nervous system that potentially could impact the superior aspect of the sympathetic trunk and produce a Horner syndrome. Or one might have damage within the nervous system itself So for example, if there were a stroke in the lateral tegmentum of the brainstem, here is a representation of the medulla, we potentially could have a Horner syndrome in conjunction with signs and symptoms that would help you localize the lesion to this particular level. In addition, one might have an injury to the cervical spinal cord, or perhaps even to the thoracic spinal cord, that might impact the ability of the hypothalamus and the brainstem to interact with the intermediolateral cell column, or one might have damage to that cell column itself and produce Horner syndrome. So of course one can have an injury to the neck and have some kind of a isolated Horner syndrome, at least isolated with respect to other neurological signs and symptoms. But whenever we have damage within the CNS itself, we're likely to have Horner syndrome together with other neurological signs and symptoms that you'll piece together as a means of localizing the injury. Well, I would suggest that we conclude this segment of our tutorial focusing on the hypothalamus with a couple of study questions. So I'll invite you to think through those study questions, and then we'll finish up on the visceromotor system and indeed all of Unit 4 by using one physiological system as a way of demonstrating how visceromotor control is integrated with sensory signals and coordinated with somatic motor control to govern behavior. And the example I'm going to choose is called micturition, or otherwise the voiding of our bladder, urination. Well, I know I've had just a little bit too much coffee this morning, so before I talk to you about voiding the bladder, well, I think I need to do it myself. So let's take a short bathroom break, and when we come back, let's talk about how the nervous system governs micturition. Well, welcome back to this final part of our tutorial on the visceromotor system. And I feel much better now that I've taken care of my full bladder. I hope you don't mind us talking frankly about this rather base physiological system, but it is an important system nevertheless. It's one that has been very important in the evolution of mammals that have used chemical signals in urine as a means for social communication, and it remains an important aspect of human behavior. In fact, so much so that I think there's some really fascinating neuroscience behind the governance of our bladder and social cognition. So I'm going to reflect upon that just a little bit as we go along. But really, my primary goal is to use the governance of the bladder and the control of the motor acts that are necessary to void urine as a way to illustrate the integrated action of the visceral motor system and the somatic motor system. Well, okay, so let's get into this session, and as we do, we will, of course, immediately once again appreciate the complexity of the brain and its extension into the spinal cord as the body's most complex organ system. And we will again be highlighting circuitry that provide for the foundation of human behavior. In this case, a very particular human behavior that I hope you will appreciate in a new way as a result of this segment of our tutorial. So my learning objective for you in this part is that I want you to be able to discuss the interplay among the sympathetic and parasympathetic divisions of the visceromotor system, together with the volitional somatic motor system in the control of micturition. Well, just to be clear, what we're talking about is urination. It's voiding the bladder, expelling urine from our bodies. Well, as I said, this isn't perhaps the most pleasant of topics to spend time together, especially given the diversity of life histories and social values and social norms that I'm sure are represented in our learning community. But again, it provides a wonderful opportunity to illustrate the operations of the central nervous system controlling human behavior. Well, let's identify the key principles that are involved in coordinating the activities of the bladder, and then we'll talk about how they are regulated in order to fill the bladder with urine, and then how to expel that urine from the bladder. So here's an illustration using the male form as our model, illustrating the various neural elements that are conveying sensory signals from the periphery and motor signals out to peripheral structures that govern this process of filling the bladder as well as voiding the bladder. Well, let's begin on the sensory side. So as we can see, there are sensory afferents that grow out from dorsal root ganglion cells that innervate the walls of the bladder. And these sensory signals are mechanosensory receptors that are sensitive to the stretch that is imposed upon the bladder as it fills with urine. There are also chemosensory elements associated with free nerve endings. Our polymodal C afferents, for example, are part of this sensory innervation to the bladder wall. So we're sensitive not just to the distension of the bladder, but also to the chemical environment within the lumen of that bladder. Well, these sensory signals then are conveyed to the appropriate sensory systems. Here illustrated here would be the anterolateral pathway that's conveying this sensory information up to integrative centers in the brainstem. Well, there are important motor elements that I want to identify for you. And let's begin with the sympathetic innervation. That sympathetic innervation is coming down from integrative centers in the brainstem and in the hypothalamus, and it reaches this intermediolateral cell column. And for control of the bladder, we're talking about the lower part of that cell column in the lower thoracic, upper lumbar segments. And the preganglionic neuron grows an axon that reaches one of these prevertebral ganglia associated with the inferior part of the pelvis. And there we find the ganglionic neuron that then supplies innervation to the bladder wall. Not shown in this illustration is additional innervation from the sympathetic ganglia to an internal sphincter muscle that is under the control of the sympathetic system. And this provides for a way to ensure bladder filling rather than bladder voiding by governing the contraction of smooth muscle at the base of the urethra that drains the bladder. Well, finally, the parasympathetic division of the visceral motor system has an important role to play in governing the bladder. The parasympathetic outflow comes from the sacral segments of the spinal cord. It is receiving input from higher brain centers, as we'll talk about in just a moment. And the preganglionic neuron, as you know, will grow a long distance. In this case, it will supply a ganglion that is very close to the bladder itself. And that ganglion will then provide innervation to the detrusor muscle, which is the principal muscle of the bladder wall. Now, let's say just a bit about the functions of these different sources of innervation. So the sympathetic innervation to the bladder is going to promote bladder filling. So it's going to relax the bladder wall. It's going to contract the internal sphincter muscle, and together that will allow the bladder to fill. The parasympathetic system, on the other hand, is going to promote the emptying of the bladder. Activation of parasympathetic inputs to the detrusor muscle will cause that muscle to contract and the bladder to void. Well, in order to actually expel the urine, we have to engage not just the visceral motor system, but the somatic motor system. And the reason is that there is a somatic motor efferent that comes from the sacral segments of the spinal cord and innervates an external sphincter muscle that's in the floor of the pelvis. And that external sphincter muscle helps to ensure the appropriate and timely release of urine from the bladder. So this external sphincter helps to ensure that urine only flows out through the urethra at the appropriate time. So in order to actually void the bladder, one needs to coordinate not just the parasympathetic outflow to the bladder wall, but also the somatic motor outflow to this external sphincter muscle. Okay, so let's see how all of this works. Let's consider first the context of bladder filling. So bladder filling is going to essentially involve an increase in sympathetic activity that will dominate parasympathetic activity. So we're sort of shifting the balance here in favor of sympathetic activity. So in order to fill the bladder, we have to relax the muscle of the bladder wall, that detrusor muscle. And so this is a beta adrenergic effect, and it allows that muscle to relax when norepinephrine is released via these ganglionic sympathetic neurons. Now, meanwhile, sympathetic activity to the internal sphincter is going to cause that internal sphincter around the base of the urethra to contract. This is an alpha adrenergic effect. So the detrusor muscle is relaxing, but the internal sphincter is contracting. So by increasing the sympathetic activity, we promote bladder filling. So what that means is that we need to relax this detrusor muscle, and we need to activate this internal sphincter, holding that urine in place within the bladder. Now, during the process of bladder filling, we also want to coordinate the activity of this somatic motor neuron that's supplying input to this external sphincter, exciting that striated muscle, causing it to contract. The more those sensory signals signal the increase in filling of the bladder, the more tone must be generated in that external sphincter in order to remain continent, in order to retain that urine within the bladder. Well, what about bladder voiding? Well, voiding the bladder means that we have to shift this balance. So now there needs to be an increase in parasympathetic activity, where parasympathetic activity now dominates sympathetic activity. So for that to happen, we need to increase the tone of the detrusor muscle. And this is mediated via the muscarinic cholinergic activation of that detrusor muscle. Meanwhile, we want to decrease the tone of that internal sphincter. And this seems to be mediated via a parasympathetic activation of the nitric oxide system. And this causes that internal sphincter muscle now to relax. But what about our somatic motor system? Well, we are not going to void urine successfully unless we have a way of decreasing the output of this somatic motor neuron to that external sphincter muscle. And indeed, this is where descending inputs come into play. There is an inhibitory interneuron that is in a position to be governed by higher brain centers and inhibit the tone of that external sphincter muscle. That muscle needs to relax in order for us to actually void urine from the bladder. Well, how is all this coordinated and governed by higher brain centers? Well, I want to walk you through this. This is an illustration of the neural systems that are engaged when we actually void urine from the bladder. So as I just outlined for you, this involves activation of the parasympathetic system, our preganglionic and then our postganglionic neurons, causing the contraction of the bladder and the relaxation of the internal sphincter muscle. But there's also this inhibition of the somatic motor neurons, which will allow that external sphincter muscle to relax and the bladder to contract and expel urine out through the urethra. Well, this is all coordinated by some really fascinating networks that involve the ventral medial forebrain and key integrative centers in the brainstem. One key center is found in the pons, and it goes by the catchy title, the pontine micturition center. So this center needs to be activated in order to coordinate the lower motor activities that result in successfully voiding urine from the bladder. So stimulation of this structure in experimental animals will produce urination, and it's on that kind of evidence, as well as an understanding of the anatomy, that we recognize this as a key center that must be engaged in order to successfully coordinate this parasympathetic outflow together with the inhibitory signals that relax the external sphincter muscle. Well, the pontine micturition center is under the influence of neurons in the periaqueductal gray matter. This is a fascinating set of cells that surround the cerebral aqueduct in the midbrain. You'll recall that there are networks of cells in this structure that are involved in producing analgesia. That is a top-down means by which we can control the transmission of pain signals in the spinal cord. There's also a center in the periaqueductal gray that can produce locomotion. So different kinds of networks are in this structure, and here's one that seems to be important in integrating inputs from yet higher brain centers and governing the output of this pontine micturition center. Well, the periaqueductal gray receives input from the hypothalamus, hopefully no surprise to you now, as well as the amygdala. And together these structures are governed by parts of the orbital and medial prefrontal cortex. Well, this is where the story of micturition, I think, gets really fascinating, because there remains in human culture still some social dimension to voiding urine. In some parts of the world, it's perfectly fine to void urine in public and to do so without embarrassment, without shame. In other cultures, in other societies, one would not think of voiding urine in public. So one can appreciate, I think, that there is a social dimension to governing the output of this periaqueductal gray in the pontine micturition center. So it's not surprising then that this network in the brainstem should be controlled by parts of the limbic forebrain that are involved in social cognition. Indeed, one of our great neuroanatomists of our time, a Dutch neuroanatomist by the name of Gerta Holstege, was bold enough to publish a paper in the scientific literature that he entitled Micturition and the Soul. And I think what Professor Holstege understands quite well is that there's a certain soulishness about the orbital and medial prefrontal cortex. And this same part of the brain that helps to define who we are as a person, as a human being, within a social context, within a social structure, is also engaged in governing some of our most basal physiological functions, including voiding urine from our bladder. So I, for one, certainly appreciate Professor Holstege's bold proclamation of relating the governance of micturition to the neurobiological basis of the human soul. Well, maybe that's another great topic to kick to the discussion forum if you wish, so I look forward to seeing your thoughts about that. Well, to wrap up our discussion of the visceral motor system, I hope you'll take away from this an appreciation that the visceral motor system operates in coordination with our somatic motor system. It's receiving sensory input, it's receiving top-down signals, even from the highest integrative centers of the forebrain. Well, I think we're ready to bring a conclusion to Unit 4 and our consideration now of our means by which the brain can control movement and mediate various dynamic aspects of human behavior. Where we're going next in the course is to step back, actually quite a bit back, and consider the development of the brain and the important mechanisms that unfold really across the lifespan that account for the changes that our brains undergo from conception all the way through advanced age. Well, finally, we're going to end up medical neuroscience by talking about human cognition. That will be the final unit of the course. Well, the good news is that I think you've had all of the anatomy that you're going to get from me. And now that we've completed our consideration of our sensory and motor systems, what remains in the course is to actually understand that anatomy in a functional context in an even deeper way. by considering its embryogenesis, its change across the lifespan, and how these sensory motor systems are integrated at the highest levels of human cognition. Well, we will along the way consider some clinical cases that will help reinforce your understanding of this functional anatomy and how neurological signs and symptoms can help you build a neuroanatomical model of the brain of the patient that might be in front of you, or the spinal cord of that patient, and allow you to localize lesions within the central nervous system based on your holistic appreciation of the function and the behavior and the experiences of that patient. Well, that's a lofty goal. We'll get there by the end of this course. And I just thought I would pause for a moment here and give you a sense of perspective as to where we are in that progression. Well, I do want to leave you in this part of our tutorial with a final study question. And hopefully that will allow you to integrate a little bit of what we've been talking about in this session and maybe prepare you for the unit quiz that hopefully you are about to embark on. And I'll see you on the other side of the quiz when we consider the changing brain in Unit 5.